So now if we really come to the, so now what is the topic of my today's lecture is different therapeutic strategy to combat coronavirus infection with a special reference to small molecules. So now all of us know what is coronavirus, and but still, for the participants, my colleagues, I would like to inform you that coronavirus is a virus which is actually a envelope virus, just like HIV virus, and it has a single positive stranded RNA, just like say HIV, and it is like. But in HIV, there are two RNA molecules, they come together, but here there is single positive strand RNA genome. It has it belongs to a subfamily Orthocoronavirinae under the family Coronaviridae, and it can be classified into four genera: alpha, beta, gamma, and delta coronaviruses. And as all of us know, so it's not an information today that what is how it infect, like how it infect is still a question not completely solved. But we can say that in last 1.5 year, we now know that coronavirus is not leaving us, and we also know that there is in several countries the third wave has already come. Although in India, government has not declared third wave, but there are all symptoms of coronavirus third waves are there, and our children are more vulnerable. Why? Because most of the, say, 60% of the, only 10 to 14% of the population is getting the say vaccinated by the double doses, and say almost 36 to percent people are having single doses, but almost 60% people are either infected or vaccinated. So we are only left, so our small children, and rather you can say the people below to 18, the 18 years of the age are remaining, say, unvaccinated or uninfected. So that is how now this opportunist virus is say attacking on these viruses, on these our small children, and their number is increasing day by day, and hospitalization of children are also increasing. If you ask Safdarjung Hospital, and if you ask the GTP hospital, they will tell you, although there is a information that most of the time it comes like that, that people say that no, no such infection, not third wave is not there, but it is there. And I can tell you that a lot of deaths are still occurring, and even in the Delhi also, I can tell you that death rate has increased, whether it is being reported or it is not being reported. But last 15 days to 1 month, things have changed. Again, it's changing, so be careful about it. And now there are 6 COVID viruses which are known till date, which are infecting the humans, and 2 alpha COVID virus, that is 229E, and the NL63, and 4 beta COVID viruses, that is the OC43, HKU1, severe acute SARS virus, and then the Middle East respiratory syndromes viruses. So these are the 6 viruses which already have infected human beings. And if you try to look into these coronaviruses, now the question arises: how do they really infect people? So there were a dialogue came on, and there were a lot of debate that is there any intermediate species there, or it is directly coming from the lab. But more or less now it is very well known that there are intermediate species, and intermediate species were mainly bat only. But these bats are being eaten either by certain animals, and you can see the pictures of the animals here, or it is also coming from the mouse and rat. But there is one major question remains here, that how it has come to, how it has traveled to different countries. Although it has traveled to different countries from humans only, but it has traveled to from such countries where there is a tendency of eating live animals. So people in Southeast Asia, in Northeast Asia, in Western countries, there is a tendency at certain countries, not in India, but in other countries, that they have a tendency to eat live animals. And it has traveled actually more and more by the live animal as compared to the dead animals or the cooked animals. Because those live animals which are eaten by the certain countries, they just freeze them and treat them so the virus doesn't die. There are certain kind of a say a certain kind of a say till date it is not known, but they have found that there are some one life cycle of the virus is surviving in that frozen animal, and that is how they are being served to the people and people eat it in the plate, live animals, and that is how that may be one major cause of communicating it to the humans. Now, they say if we really want to understand the life cycle of the virus, we can say, as you can see here, that it is shown here that there is a virus that enters to the bat, then bat releases it, and then that is how then it reaches to an intermediate host animals. And there is a question mark here, domestic animal. So there were in Singapore and several other countries, they have actually what happened in India, people have not say sequenced the animals, they have not checked animals, they have not done much testing on the animals, but it has been found that there are certain in the certain part of the country the animals were also affected. But as in Singapore and other countries, they have clearly shown that their cats were infected, their dogs were, monkeys were infected, chimpanzees were infected, and even the infection has traveled to the zoos also, in certain zoo. So it was came out as maybe the intermediate host is animal, but there are information that maybe bat is directly putting it into the humans. And now the question arises that what is the virus, what are the different proteins in the virus, and what is the structure of the virus. So if I go directly here, that will be more clear picture here, that viral RNA has it has a trimeric spike protein, it has a, say, a membrane protein, which is having this kind of a structure, then it has an envelope protein, it has a nucleocapsid protein. And if you try to understand it, when we say spike protein, so spike protein actually it attached to the host receptor ACE2, and ACE2 receptors are found in the human being in different organs. It's not people only very commonly know about its occurrence in the on the heart only, because ACE2 receptor were very well known for the treatment of the heart, but it is found in almost several organs of the human beings, and that is why it is very easier for the coronavirus to attach itself through to the ACE protein, through the S protein. S spike protein of the virus attaches with the ACE2 receptor of the humans, and ACE2 receptors are available in the nasopharyngeal region and in the nasal region in enough quantity, and they can that is how they, most of the time it has been found, they enter, the virus enters inside the body through ACE2 by attaching itself to the ACE2 receptor, and then after attaching to it, they have a different life cycle, which I'll tell you. And there is a kind of a M protein, which is called as the membrane protein also. Then there is the N and E protein, which I've already told you. And hello. मैं तुम्हें ना बाद में कॉल करूँ, I'm in a meeting. So when there is now what happened that when you have so there are other still there are some questions, and I'll show you that other other targets are also there, and other proteins are also there which are responsible for spreading the infection inside the viruses. And now the question arises that these are the part of the virus. Now, what are the host factors which are responsible for the infection? So one I told you, that is the ACE2. Then they have also find that there are some other say proteins and other say mechanism of the infection are also there, and those mechanism of infections are uh, now the question arises because everybody says that in the that elderly are more susceptible to this viral infection, and people who are suffering with some kind of a respiratory infection are more susceptible. People who are having the diabetes are more susceptible. Why it is like that, and why it is being said? It is being said like that because the people who are say for example, if you are having everybody has two kind of immunity. One is innate immunity and another is the adaptive immunity. And innate immunity is found mostly in the infants and up to certain age in the human being, but after that it converts into the adaptive immunity. And the innate immunity and the adaptive immunity is responsible for saving you from any kind of a say pathogen or any kind of an infection to you. Now what happened that, as all of you know, and maybe if I'll find in the time in the due course, I'll tell you also that we know that there is a word comes in the say in kind of a COVID virus that the after the fourth and fifth day of the infection or sixth day of the infection, there is a say there is a uh, infection storm occur, or you can say there is a storm of say is there is a kind of a inflammatory response in the body, and there is a kind of a cytokine storm occurs in your body. Now question arises that if there is a cytokine storm, if there is an inflammatory responses, we should be happy because this is a normal inflammatory response should occur in anybody if there is an infection. Then why are we so worried, and why are we saying that cytokine storm or that inflammatory response is actually killing the human being? But the kind of the responses and the kind of cytokine storm is occurring in the response of the SARS-CoV-2 virus is different. Why it is different? It is different means there is an infection, and what occurs that the virus is so smart that it induces the in our body there are some 36 kind of cytokines are found. 16 are basic, and then some classified cytokines, then some subclass of cytokines are also there. So these kind of cytokines are, all of them, they get inflamed. And what inflamed means, they get their secretion and their some of them are secretory cytokines, some of them are non-secretory cytokines. It has been found that virus sends some such kind of a message through our brain to these cytokines that their generation increases a lot, whatever is required, more than that. And they start engulfing the normal cells, they start engulfing the normal cells of the body rather than engulfing the viral cells. So that is how what happened, that there is a so this kind of a response, we have to stop these responses. So it has been found that, and there is a lot of say discussion is going on that can, why everybody doesn't have a response, why out of say 100% patient, if we take 20 patient, why only in 5 or 6 patient this kind of a cytokine storm occur. So they have found that this kind of a cytokine storm, now question arises whether this cytokine storm is because of the certain strains only causing these kind of a cytokine storm, or there are certain people who are more susceptible for these diseases and they are undergoing these cytokine, these kind of say answers are still not there. People still don't have these answers that whether cytokine storm is because of a certain particular COVID strain or because of a particular person. because a person is more susceptible, or a person is having an innate immunity, is having the adaptive immunity, low adaptive immunity, that is why it is occurring. So on the basis of certain clinical trials, certain clinical studies, they have found that the people who have lower immunity in their body, they are suffering more with these kind of a drugs, with these kind of a say disease further. But the question arises that if you really want to handle these viral infection, you have to be given the certain kind of a steroids. And it has been found that when the steroids were given more, not in the regulated manner, then your immunity goes down, and then people who are being attacked by the cytokine storm, they die immediately within few hours. So it is, you need to a balance between the treatment modality, treatment strategy. You should, so you have to treat a person to person in a symptomatic manner into so what happened initially, that when clinician also didn't understand this, they did not have time to understand this disease, so they started treating people with same yardstick. And then it has happened that people who have been treated for the steroid for a very long time, their immunity gone down, and they got certain kind of a fungal infection, and they died because of the they some other secondary infection rather than the COVID also. So it took some time to clinicians, and actually by the second wave only they could understand this virus more clearly. And when the viral genome was sequenced, then also they could understand it more clearly. And by the time, lot of human loss was already there. So what they have found by now, that people who are suffering with hypertension, chronic obstructive pulmonary disease, diabetes, and cardiovascular disease, they become more more susceptible to this particular virus. And what are the severe complications which occur because of this medicine, because of certain this virus? That is, you have a respiratory distress syndrome, you may have a septic shock, metabolic acidosis, hard to correct, coagulation dysfunction, and multiple organ failure. So I don't have time to tell you about all these things. You can one by one go to the say certain on the WHO website. I will suggest, please don't read from the Google. Please the COVID website of the WHO. You will find every small detail about the COVID on that there, and that is a certified detail. They even have uploaded so many lectures, so many studies, so many papers, and so many reports in the simple language. You need not to be a graduate in biology, you need not to be a very well informed person. The only thing, if you know English, you can read it. So I would suggest you that read it from the WHO, read it from the WHO report. There are several WHO report and several reports on the ICMR website. They are in Hindi also. You can read it and you can understand it, that what it is. So now if I go to the life cycle of this virus, because one should always understand that what is the life cycle of the virus. If you don't understand the life cycle of virus, you cannot treat a disease. So you must know that how it enters. So as I told you, that it may enter, there is this is a cell wall, cell membrane of the mammalian cell, and then there is a say virus comes, it get through its spike protein, it connects it with the ACE2 receptor. It can also go by the simple endocytosis also through certain ion channels. It can enter, then it can enter through so because it is a small virus, so what virus does, that it releases its RNA, and iron enters through some in the our membrane. There are certain pores there, so it can simply by the endocytosis, it can enter inside the cell, then it goes and binds itself to the RNA, and then it translates the through the say mRNA and gRNA, and then after translation it first it makes the non-structural protein, and these non-structural protein along with the RNA. This is the normal way of producing a any say viral protein inside the body. So this is a normal way. First the translation, then the transcription, and after the translation and transcription, after translation and transcription, you will find that the mRNA of the virus will be formed, and the different proteins, spike protein and say nuclear protein and membrane proteins are found. Then they further translate, then the structural protein synthesis occur. Then these all proteins and envelope, they assemble, and then after that they make a whole virion particle, and this virion particle, through exocytosis, released in the host cell, and then it infects the other cells. So that is how only the mammalian cell act as a factory for the producing the different COVID virus inside our body. And that is what is the most sad part with the viruses, that they utilize our own machinery, our own say all kind of energy, glucose, everything, ATP, and that is how they convert themselves. They divide and divide, and they infect more and more number of cells and more people inside our body. So now I'm coming directly to the proposed therapy, because my talk is supposed to be on the therapy. So if I talk about the therapy, so there are, now if you look at this slide, it is little bit confusing with you, but uh, in order to summarize my point, I have kept it here. Now question arises that the how would you really find a drug for the virus, and how should you really control it? For example, fine, we have today so many vaccines, we are vaccinating people, and even in the vaccine also we have challenges, and we all of us know that lot of say new say strains of viruses are coming, where the mutations are occurring, delta virus, delta mutation, beta mutations, and purposely government of all over the world, WHO have given them a name. Why they are giving one by one them a name? Because they want that everybody should understand what is alpha mutation, what is beta mutation, what is delta mutation, because it is because they wanted that that the viruses with the different mutations can be identified as, because if we will not classify them, we will never come to know what is happening to certain people and what is happening to the other people. Why some of them have the severe diseases, why most of them have not a severe diseases, because still it is a question that why some of them are having the severe diseases. As I told you, it can be a person to person, it can be a particular person there, or it can be strain also. So, and I tell you, there are certain information. If you read very recent data on WHO, they have found even that the people who are suffering with the Delta variant also, some of the people who are suffering with the Delta variant also, disease is not severe. So what is happening actually? Why it is happening? And because you don't understand it, so you need to understand every part of it before treating or before giving a therapeutic modality. So now the question arises that they have the whole sequence of the virus, whole genome sequence of the virus. Now they have the transcriptome sequence also of the virus, this particular virus, and that has shown that it is similar to the SARS virus 1, and but it is not completely homologous to the SARS virus 1. So they have taken some lesson from the SARS virus 1, and they have taken those strategies which were taken in the SARS virus 1, and they have started their strategies, whether it is vaccination strategy or it is their therapeutic strategies. They have learned from there and they have used it. So that is why it is told always that learning is never useless. Say people were not much worried in the SARS-1 because it has infected only the Asian countries. Very less Western countries or rich countries Western countries were infected. So people did not make so much of hue and cry at that time. It was lethal. It has caused a lot of deaths. It has caused millions of the deaths at that time also. But less number of rich people died at that time, so people did not worry. But somewhere our scientists were studying that, they were creating some kind of a, they knew that there is something new will come. Virologists have told it to the world that COVID, that there will SARS-2 will come. People knew that SARS-3 will also come. They know it. That there will some such infections will come, and so that is why what they did actually, that they took the SARS-CoV genome, and then they take the human genome, and they have done, especially through some computational studies, they have found a network between the human protein and the COVID protein, and that is how they have find out a kind of an interactome. Means they have given us an information that certain proteins of COVID are interacting with the certain proteins of the humans. So they thought, okay, if there is a protein which is interacting with the SARS-CoV virus, now if we know that this protein can be regulated by a particular drug, so they directly find out those small molecules and those drugs, and they said, okay, we can use these particular drugs for handling the viruses, for reducing the infection of the viruses. Let me tell you one thing. There is a misconception between the people that if you are infected by something and I will give you certain drug, you will have no further virus in your body. It never happens. Fine. You will, if once you are infected, you will remain infected for a certain period. There are very, very less chances that the viral load will be zero. Fine. So always keep it in your mind that you are, if once you are infected, or even if you are virus load is not coming, and whenever you are going for RT-PCR, you are finding your test zero, and you are finding you have no infection, it is wrong. You are infected, you are having such kind of infection, so you should be very, very much sure about it. So that was the first strategy that they started finding is drugs from here. The second strategy was network based drug repurposing. Now the question arises that, okay, the first strategies was that they came to know certain mammalian proteins, they came to know certain host proteins, and those against those host protein drugs were already known. So they thought that, okay, we will use those particular drugs and we will save people. Then there were another kind of a strategy came where they came to know that there is a network based drug repurposing. What is this? There is like, when they come to know that there are certain proteins are there, and they came to know that that this protein has certain role in diabetes, this protein has certain role in pneumonia, this protein has certain role, this receptor has certain role in say influenza virus, this protein has certain role in HIV virus infection, in Ebola virus infection, in say any other. So they thought, okay, against this protein, we already have a drug. Why don't we repurpose, reassign this particular drug against the HIV, against the SARS virus? So that is what they started another strategy. Third strategy was that the which is called as the HCOV induced transcriptome. So what they say, and then drug induced transcriptome. So whenever they took they did two kind of studies there, one in the plate and another was directly in the human being. So people who were infected, they took the mammalian cell, they grow it in the in the petri dish, and then they infected it with the virus, and then they took this particular mammalian cell and they did the transcriptome analysis using the next generation sequencing. So they did the gene analysis and they found which particular genes are being upregulated, which particular genes are being downregulated, and then they did the same studies by taking the blood from and say tissues, or from the say plasma from the, or maybe cells from the infected people, and then they merged the lab study and the human patient study, and they merged the transcriptome, and they found a whole signature of the genes which are being upregulated by the and the downregulated by the infection. And then what they did, then they took the treated people also, people who have been treated by the certain drugs with the infection. Then they did this work in the out of the people who have been infected and the people, and they did in their laboratory also. And then they again they did the transcriptome analysis, and they found the genes. Now when they treated and when they non-treated, they compared it. And by treatment and non-treatment, they did the enrichment analysis. What do you mean by enrichment analysis? They saw that there must be certain genes which were upregulated in the untreated person, but when you treated them, they have come to the normal axis. And when there were some downregulated genes, they also came to the there. But there were certain genes which were having no effect by the drug treatment, or there are certain genes which are got upregulated and downregulated because of the drug induced gene. So they did a big enrichment analysis, and then by the enrichment analysis between the genes treated and untreated, they did a network based prediction of the drug combination. Means they said, okay, you treated somebody with the remdesivir or the hydroxychloroquine, but that patient did not survive, or the patient could not be cured, or the infection did not go down because these particular genes were not normalized. When you treat it with a non-infected person, when you compare it with a non-infected person, you found that by the network-based prediction studies, they found that the one drug is not enough, or the two drug was not enough. You need a combination of drug to normalize the transcriptome level of a particular person. Then they have given a kind of a drug combination. They suggested a combination of the drug which can be used for the treatment of the people, which is being, say, I'm simplifying and telling you here, but it is little complex, but just to make you understand, because all of you may not be from the virology background or the drug discovery background. So I just wanted to inform you so that you can inform your student and you can teach them there. So I can share this presentation with Anshika, and she can always share it with you also. So you can, then what happened that when you have a network based prediction of the drug combination, now what happened that by these combination studies, and then this, what is this sixth, the last one is saying that when the coronavirus infected people, okay, and they wanted to see it that once they infected and they have isolated those viruses from the humans, and then again they took those viruses and they tried to see that are those treated viruses are developing some kind of a mutation because of the treatment, or these viruses are getting getting mutated because of the drug treatment. Because this happens in HIV. All of you know that when the raltegravir, elvitegravir, lot of drugs were given AZT, then there were certain mutations occur in those target proteins because of the treatment. Sorry, excuse me. So when they treated these particular people, they further take out, and they, after taking out, they saw that whether these treated people are having certain kind of a mutation, then again they created a network in against those mutated virus, and they have again by prediction they suggested that okay, drug combination can be changed because this drug is causing certain kind of a mutation, so let us not treat with this particular drug, or we can reduce the reduce the dose of these drugs. Because let me tell you, I don't know whether you people know the resistance pattern or not. It is known now that you can reduce the resistance by the persistence. There is a difference between the persistent cell and the resistant cell. There are resistant cell is that if you treat, if there is a resistant cell is there, even if you increase the dose of the drug up to 10 times, 10, 20 times, they will not die. But if you treat if there is a persistent cell, you will find that there is this persistent cell can be taken care of, can be killed if you increase the dose of the drug. So people always suggest you that please take antibiotic, take antiviral drug for a certain time and stop it. Don't take it forever. Okay, don't take it for a very long time. So there are chances that you will create a persistent cell, and if you create a persistent cell, then the resistance will go not go like this. You will have a plateau. There will be a increase in the dose, then there will be a plateau, and after very long time a resistance will be there. Fine. So if you really want to do that, so that is why they suggested that, okay, drop this drug, include this drug, reduce this drug. So dosing pattern is also important. So this whole slide is very important for any kind of a therapeutic strategy, but specifically for the SARS virus. So if you look into the strategy, you can always see that we can treat with the vaccine, we can treat with the IgG immunoglobulin, which is called as the biological antiviral drug. Then the prophylaxis strategies can be and the repurposing of the drug. What is this prophylaxis strategy? Rest, I think you people can understand. Prophylaxis strategy is that you are eating some particular molecule, even if you are not infected, you are being given it in advance so that the chances of getting infection will be less to you. So all frontline worker, all say government worker, all health worker, doctor were given certain drugs under the prophylaxis strategies. And it is unfortunate to say that certain people also started taking this prophylaxis drugs. You should have given it, people who are suffering with diabetes or certain chronic disease should have taken it, or the people who are having the age more than 60 or 65 should have taken it. But in India, a lot of people have started taking hydroxychloroquine or some this homeopathic diseases or some immunoboosters or something. I won't say it is the right way of taking it, because you never know what it is doing to you in your body. So until unless we should leave certain things with the clinicians, we should not become doctor ourselves, because we are I tell you, even if I pursue science from last 20, 30 years, but when we sit in front of a clinician, we have no knowledge. So the now what was the problem when we tried to do it? Now these problems actually doesn't make any sense. When I made these slides, it made some sense because there was no antiviral assay at that time. So how would you really develop a drug and check it that whether it is actually inhibiting the drug or not, inhibiting the virus or not? So at that time there was no assay. There was no pseudovirus particle was there. What do you mean by pseudovirus particle? You cannot handle, everybody cannot have a BSL-3 facility and have the viral facility in your lab. And if you will try to do the science in the viral facility, then everybody cannot do the science. The total number of people who are doing the virus antiviral work will be very small. So in every viral research, they develop certain kind of a pseudovirus where it become a non-infectious virus, and you can handle them in the lab without any specialized facility. You can have just a cell culture lab and you can work with that. So they didn't have any such thing. They did not have any no recombinant protein. There was no spike protein with us. There was no membrane protein with us. So even if we want to do some kind of a study, how will we do it? You want to check that how it is working, what is the function of this protein. You have no protein in your hands. And everybody cannot go to the virus and grow it and then isolate the protein, and then you cannot do the research. So no animal model was there. So for preclinical studies, you need animal model. But what the good thing was there, that Johnson and Johnson has frozen a strain of a transgenic mice with them, and that for which they developed for the SARS virus 1, and they just took out that from their freezers and they started developing the transgenic mice. Why do you need a transgenic mice model? Why do you need a special model for this treatment of this virus? Let me tell you, there was a problem, and I think one of my slide was there, but I did not explain you. The problem is this, that every mouse, every rat, or every kind of a, say, our model, clinical model, which we use, a small animal model, also have the ACE2 protein. So now question arises, when they themselves have their own ACE2 protein, but you want to turn it, check it for the human proteins, human ACE2 virus, human ACE2 protein, you have to make a mouse where their ACE2 is silenced, but your ACE2 is being, say, expressed. So you need, so that is why you have to make a transgenic mice where you have to introduce the human ACE2 there, so that you can get the results in terms of the human, what this drug or what this vaccine will do in humans as compared to the mouse. So that was the very thing, and I would say that all these problems have reduced, but still government is funding a lot for antiviral assay, for pseudovirus particle, recombinant protein production, transgenic mice and transgenic animal production. Not only this kind of a transgenic mice, another mice model. If you can develop a mice model, because I tell you, for one particular molecule, THSTI and RCB is charging you around 1 lakh to give you a result that your molecule is active or not. And I find it is very unreasonable, because they are the government agency, but if you go to them for primary tests, they're asking 25,000, and for the final result they're asking 1 to 1.5 lakh, and which is not acceptable, at least to me. So I think we still need more and more assays, more and more model, so that there is no monopoly for the agencies or the industries which do the drug testing. So I'm not going to talk about the vaccine candidate, because I will more and more I will focus on the drug candidate, because by vaccine you people know much, and you know that a lot of vaccines are in the market, you are being treated, and if there will be any question after my lecture, I can take it. So now if we so now question arises that what were the, my lecture is up to 12 o'clock, no? I'm
Yes,
asking.
ma'am. Yes, ma'am.
Okay.
Ma'am, you can continue a bit more.
So
You.
I will go up to the 10 minutes to 12. Fine. So there
Okay.
are certain drug candidates which were proposed, and those lopinavir, ritonavir, remdesivir, erbidol, cam, lot of drugs were, say, maybe in India you were only you only heard about hydroxychloroquine, remdesivir, some famous drugs came to know, but every country was using different kind of drugs and trying different combinations, and they have found the target candidates also, and all of them are actually repurposed drug, means they were being used for other viruses, other diseases, and they have been repurposed for the SARS-CoV-2. So, and their target in the SARS-CoV-2 are 3CL-Pro. This is one of the protease enzyme in the say coronavirus, then PL-Pro, this is another protein, then RNA-dependent RNA polymerase. This is one of the first good target which was used. Then this S protein, then TMPRSS2, then ACE2. So all these angiotensin converting enzyme, transmembrane serine protease. So they have find out different drug candidates against these different targets in the virus. Then there are some more drug candidates were there, baricitinib, ruxolitinib. When you say IB means it is a antibody, humanized antibody conjugate, which is being used as a drug candidate. So these are few, these are not a small molecule, these are the small. So what they did, that these were the already known JAK inhibitor. JAK is a kinase which is known, which is actually used for the targeting the rheumatoid arthritis or myelofibrosis. They repurposed it for the COVID-2, and it has shown some very good responses also in certain candidates. then certain patients, then lopina, we read it. Then another very important molecule, favipiravir. Favipiravir is a targeting the RNA-dependent RNA polymerase. Then remdesivir, then arabidol, chloroquine, it became very, very famous, which was being used. Then chloroquine, another analog of chloroquine, which is known as hydroxychloroquine, that was also infecting the malarial parasite. That initially showed very good promise, and specifically for the prophylactic strategy. And almost government in India has given hydroxychloroquine to everybody. India has given it to several countries free of cost, because in India the hydroxychloroquine production cost is just 10 paisa, 10 to 15 paisa. So they gave millions and millions of hydroxychloroquine to other countries. That was a very good gesture of our government. And then these are the certain drugs which were in pipeline. I shouldn't say which are in pipeline, but because this pipeline is changing every day. Fine. Now, there was a say SOP of the government of India, or there was a line of treatment which was suggested by the WHO. Now hydroxychloroquine and remdesivir are now dropped completely from those SOP, and they said that they are not causing any benefit to the person. But I tell you, remdesivir is being used in India in several hospitals, because they say that after COVID, the secondary lung infection, it is actually helping people. But government has said that please don't give it to the people, but clinicians are giving it later on. Then baricitinib, ruxolitinib, favipiravir, ribavirin. So number of compounds were given for the treatment of this. So I'll not discuss hydroxychloroquine because it is not no use now. People are not using it. I will go rather to certain molecules which are being used today. So the question arises, who gave them idea that remdesivir will work? So there is a big company, Gilead Sciences. Gilead Sciences is very, very famous company which does lot of research on the viral SARS viruses, specifically on the SARS viruses. And Gilead was already working on this project of production of remdesivir, and from there they have immediately, within a month or two, they showed that remdesivir is working and it is binding to the RNA dependent polymerase. You can see here in my slide, then this is the protein of RNA dependent RNA polymerase, and in a small pocket remdesivir is going and binding. And then they said that you can see that here again, this is the remdesivir, and these are the amino acids molecule of the RDRP, and they showed it that it is working effectively, and that is how they started working it. Then there was another molecule, baricitinib, which is, I told you, is a JAK inhibitor. And baricitinib actually it's a known molecule. It was a known molecule, and it was being used for the rheumatoid arthritis. And so what they did actually, that they, and please look at the molecule. Molecule is not a big molecule. It's a small molecule. It is a pyrimidine kind of a molecule. It's a kind of a nucleic acid base here, and pyrimidine with a viral-like structure. So it's a kind of a nucleic acid base. Then it has a another 5-membered ring. So you can see it's a small molecule, and this particular molecule actually act on the JAK kinase, and how it acts and how baricitinib work actually. So there is a virus here. Virus binds to the ACE2 receptor of the host. Then it gets expressed on the AT2 link cells, and then this ACE2 inhibitor or virus can also enter through the endocytosis. Viral entry can occur through endocytosis also. And now what happens, as soon as the endocytosis occurs, there is a kinase which is known as the AAK kinase. So this AAK kinase is actually, so I'll read it from here, that so that you can understand what is written here, that it is queried by the fleet of algorithms to identify new relationships to suggest new ways of tackling disease. AAK1 associated kinase is actually in fact the cyclin associated kinase, which is called as the JAK kinase. So what does it mean actually? That there are like AAK is being upregulated because of this ACE2, because of the infection of the virus. So if you want to inhibit this AAK upregulation, if you want to reduce the upregulation of AAQ2, if you treat by the baricitinib, erlotinib, sunitinib, and fedratinib, they actually, what they do, they inhibit the AAK1 expression, and that is how and they also bind to the GAK, and they, what they do, that they stop the endocytosis, they stop the ACE2 mediated signaling, and that is how they reduce the infection of virus inside the body. It is also known that baricitinib directly inhibits the JAK2 also, and by inhibiting the JAK2, because JAK2 is involved in the cytokine signaling, so it reduces the JAK2 when JAK2 is inhibited, then the cytokine signaling is also inhibited and the inflammation is reduced. So it also reduces the cytokine storm. Now question arises, we need cytokine response, but we don't need it over and above what is not required. So you have to give baricitinib at a certain stage. That is what I'm telling you from very beginning, that there was a problem while the treatment of the COVID was being done. You cannot give somebody baricitinib on the first day. You have to give baricitinib to a COVID infected person on the 4th and 5th day. Don't give it initially, otherwise your immune response will go down. So that is what was the problem actually, that the treatment modality was not defined, and that is why so many mistakes occurred by the clinician in treating the people and handling them. Then another molecule which came in the picture was the ruxolitinib. So ruxolitinib is also a molecule which was actually given. This is the structure of ruxolitinib, very small changes than this, and I have already told you how it works. But here, the primary target of ruxolitinib is the JAK-STAT pathway. JAK-STAT pathway is one of the pathway, a signaling pathway in our body, and that when it affects the JAK-STAT pathway, and why JAK-STAT pathway is important for us, because JAK-STAT pathway is also responsible for the secretion of interleukins or the expression of interleukins. What are interleukins? Interleukins are the cytokines only. So they are responsible for the cytokine secretion and cytokine treatment. And actually, ruxolitinib was a molecule which was used for the treatment of blood cancer, but they tried it. To dampen the cytokine storm. They have the Insight and Novartis did the phase 3 clinical trial with it, and they have said that this is a successful molecule to reduce the cytokine storm in the viral infected people. Then there is another molecule which is called as the favipiravir, and favipiravir is also being used, was already being used against the influenza virus, and they were already using it. They already also tried it in the phase 3 clinical trial in Japan and phase 2 clinical trial in US, and they said that it acts on the RNA polymerase and it reduces the viral replication, and they are still using favipiravir for the treatment of the COVID infection people. Then ribavirin was used largely in the Canada, and it was it was already being used for the treatment of hepatitis, and that was the molecule which was inhibiting the inosine monophosphate dehydrogenase. So the inosine monophosphate dehydrogenase, they have shown that this particular protein has similar structure as the PL-Pro, one of the protease of the SARS virus was having. So by superimposing their structure, they thought that if they treat with this drug, then they can inhibit the protease enzyme, and they can actually say manage the virus infection. So it also inhibits the viral replication inside the humans. So these are the few drugs which I have told you. It's a very long, very long presentation, but I just wanted to tell you few of the very new structures which were given there, and there is a huge list. So I will just give you one or two important molecule which has given you some. So I just want to tell you here that let me bring you here. Fine. So there is in HIV, Merck has made a lot of money against HIV, against certain drugs which are there is a protein which is called as integrase, and as its name suggests you, integrase is a protein which integrates the mammalian DNA with the viral RNA. So what it does, it's such a smart protein. It prepares viral RNA, it cuts viral RNA, it does the strand exchange. It brings it from that viral RNA inside the bacterial inside the human cell. Then it cuts it. Then it conjugates it with the viral DNA. Then it does the strand exchange also, just like any replicating enzyme. Because in replication there is a step where there is a cutting and then ligation. So it works as a ligase enzyme. It cuts it, then it's ligated with the viral DNA, then it does the strand exchange. Why it does the strand exchange? If the strand exchange is done immediately, then what will happen? That there will be a heterogeneity will be there. Means you will be able immediately the viral DNA will be exchanged between the two strands. So so it will be spread immediately. So what they found, that they have developed a drug, raltegravir, and its analogs were further developed. You can see its structure here, elbitagravir and dolutegravir, which can stop the integrase activity. Means which can inhibit the integrase function of the conjugating the viral RNA with the human DNA. So they use the same drug they thought that they will use the same drugs for SARS-CoV-2, and there is a protein in SARS-CoV-2, 3C, and it has shown very good activity, but it has not reached to the clinical trial in the SARS-CoV-2. Then they have again tried to use another molecule which was actually bind to the CCR5 receptor. And CCR receptors are actually known for causing the infection in the human beings. So they have taken a drug which is known as the Maravirac. And Maravirac is one of the very successful drug here, known against the HIV. And they took this CCR5 receptor binding drug. And when they took this drug, they found that this particular drug also recognizes the S2 subunit of the spike protein. So they thought that if they will use this particular drug and some other drugs like enfuviratide, which binds to the GP41 of the HIV. Then they thought that if they can take a palivizumab, which also binds to the RSV glycoprotein of the RSV. RSV is another Rous sarcoma virus. It's another virus. So they found that these molecules have similar, the molecules which are binding to these proteins in different viruses, they have a very good affinity, but with the S2 subunit of the spike protein of the SARS-CoV virus. So the lot of work has been done and lot of clinical trials were done with these molecules against the SARS-CoV-2, and still data has to come that whether we can use it or we cannot use it for the SARS-CoV-2. So similarly, there are other drugs like acyclovir, which is actually a say HSV1 thymidine kinase inhibitor. So they were then there are certain, and you can see they're just a small molecule, very small structures, not a very great structures, but they do wonders in the as an antiviral drugs. And there is one thing which I would like to share you, that most of the time you'll find the antiviral drugs which have been used till date are nucleoside drugs, because whenever you use a drug which is having the nucleic acid basis, the chances of having a kind of a inflammatory response will be less, because they have the similar structures in the DNA. So you will find that the acceptance rate will be much better as compared to the any other molecule. So that is why most of the time in the viral diseases, these kind of a nucleic acid or nucleoside drugs are being used. So similarly, then there are some HCV against the hepatitis. There was a number of drugs were used, and hepatitis A, B, it was fine, but C, D, E are actually fatal to the people, and most of the people actually they don't know that people who suffer with the hepatitis E or hepatitis C or D, they are they like they become severely ill, and sometimes you will find that they can never go back to the virus, they can never go to the normal life. And people who suffer with the hepatitis say hepatitis, they can, after some time they develop the cancer also, hepatic cancer also. So that is why it is one has to be very careful if you have any kind of hepatitic infection. So then there is a leptis, there are these kind of infections are there. And so then again, these kind of a molecule, embitisavir, velpadasivir, elbasivir, these certain molecules were again tried against the against the SARS-CoV viruses. Then there were certain disease, there were certain drugs, beclabuvir, desabuvir, then sofosbuvir, that was being used against the 2M-Pro protein. I told you about that. This is a membrane protease protein of the SARS-CoV-2, and they have done some kind of computational studies, and then then other studies, and they have published in this journal the results of these bindings and these wet lab studies. And they found in some part of some countries, they have found that it has beclabuvir has given them some very good results in some of the COVID infected patients. Similarly, there are now more or less every study could not be converted into the wet lab studies. Most of these studies, which I will show after this slides, are only the computational studies. The wet lab studies are slowly coming, which I have not included there. So then they have found that there was in influenza virus, there was a portion where a amnitidine drug used to go and bind. So they tried to use this drug against the SARS-CoV-2 virus. Then similarly, they have again found that there was a drug, Zamnavir, and there was a drug, Leninaviril. So there were certain molecules which were binding to the influenza virus, RNA polymerase of the influenza virus. They tried because there was a lot of homology between the RNA polymerase of influenza virus and SARS virus. So they tried these drugs on the SARS RNA polymerases also, but the wet lab results, I don't have it here. So similarly, they tried with the interferon alpha and interferon gamma also, and they found there were certain molecule, podophylamide, catechin. Catechin is a very common molecule, and they found that they have shown certain kind of activity in the SARS-CoV-2 virus infection. So you can say that you can work in three ways to treat the COVID-2. I'm just finishing in after this slide. One, you can block the viral replication. Second, you can prevent entry into the cells. And third is that you can reduce the hyperimmune response and acute respiratory distress. So at the all three stages, one need to tackle this COVID virus, that you have to if once you have to first prevent its entry into the human being. Second, you have to, if it has entered, you have to stop its replication. Third, whatever is the secondary effect out of this drug, you have to reduce it. You have to reduce the hyperimmune response and acute respiratory distress. You have to save the lungs of the people. You have to save the person from the secondary infection. And that is how, these are the three major ways to treat the COVID-19. Thank all of you for listening.